Chapter 12, Nervous Tissue. Overview of the nervous system. We can break up the nervous system into two distinct parts, the central nervous system, or the CNS, which includes the brain and the spinal cord. The peripheral nervous system, or the PNS, which includes the cranial nerves, spinal nerves, the enteric plexuses in the small intestines, and the sensory receptors in the integument system, or the skin. This flowchart gives us the organization of the nervous system, specifically the central nervous system. So we see on the left over here our sensory division. So with our sensory division, we deal with two types of senses. One, general senses and special senses. So we talk about somatic sensation and special senses. And when we talk about special senses, we're dealing with our 5 special senses: hearing, balance, taste, touch, and vision. Our somatic senses would be anything to our skin, like pain, temperature, pressure, tickle, itch, things like that. So that's our sensory input. That's the input coming in from the outside environment, stimulus coming in from the outside environment and moving its way into the central nervous system. Now, from our motor output, so once the central nervous system decides what that input is and how to respond, it can go one of a couple branches. So if we go to our first branch, we deal with the somatic nervous system. This is all motor output to skeletal muscle. So if you were to give an example of this, we could talk about reflexes. If you were to touch a hot stove, that heat would be detected and subsequently pain. It moves to the central nervous system, and the central nervous system will go to our skeletal muscle and say, move away from that stimulus, and you get a flexor reflex. So it flexes the forearm to move the hand away from that pain. The second branch is the autonomic nervous system. So autonomic means self-guided. So this portion of the nervous system is broken up into 2 major divisions and a third as well. The 2 major ones are, you probably have heard of this, fight or flight, which is our sympathetic nervous system. This touches smooth muscle, cardiac muscle, and glands. Now, the parasympathetic is its foil. It is the rest, digest, repair, reproduction portion of the autonomic nervous system. Again, you notice that they're touching the same thing. So smooth muscle, cardiac muscle, and glands. The last portion of the autonomic nervous system is the enteric nervous system. This is handling all output, motor output to smooth muscles and glands of the gastrointestinal tract, specifically digestion. Functions of the nervous system. So we have 3 major functions of the nervous system. One includes sensory, what we just talked about. Its senses change through those sensory receptors. Motor response, responding to that stimuli that was detected from those sensory receptors. And then an integrative function, so being able to analyze, integrate that incoming sensory information, store some aspects of it, and then make decisions regarding appropriate behaviors and response via the motor pathway. Our next subset is going to look at the anatomy and histology of nervous tissue. So we're going to start with neurons. This is a classic multipolar neuron. How do we know it's a multipolar neuron? Well, we look here at our cell body and we see that we have these dendritic spines coming off it and an axon. So that gives us 1, 2, 3, 4, 5, 6, 7, 8, more than 2 processes or projections off of that cell body gives us a multipolar neuron. Now, how do we know, or what are some functions and characteristics of neurons? Well, one, they're electrically excitable, meaning that they can process through a range of states, being depolarized, repolarized, and even potentially hyperpolarized. So we're talking about shifting ions, shifting charges in and out of this cell. And we'll look at cellular structures. Structural classification of neurons. So we already on our last slide outlined our first one, which is the multipolar neuron. So neurons can be classified based on the number of processes extending from the cell. Again, with all those dendrites and the axon, this gives us more than 2, so it is a multipolar neuron. Now, these are the most common neurons found especially in the central nervous system. We're going to be multipolar. Next are going to be bipolar. These are going to be relatively rare. We look at the 1, 2 extensions off the cell body. So this is a bipolar neuron. Well, they're going to find these when we do special senses in the retina. The third and last classification of neurons is a unipolar. So again, cell body with one process off it. We'll find these typically in our sensory pathway. So taking that stimulus, moving it through the peripheral nervous system to the central nervous system. So that pathway coming into the central nervous system would typically be made up of these unipolar sensory neurons. Let's look at examples of dendritic branching. So we deal with two types, Purkinje cells and pyramidal cells. We're going to find Purkinje cells localized and specialized in the cerebellum. Now why? Why are these branches important? The branching allows dendrites to make new synaptic connections. Therefore, this extensive branching can be considered a sign of complexity because of the increased number of synaptic connections it allows for. The next step is looking at functional classification of neurons. So we look at structural, now we continue with functional. Neurons can be classified based on the direction of nerve impulses and the way they propagate through that nerve. We can look at sensory neurons, and we already said that most of our sensory are going to be unipolar neurons. These are called afferent neurons because they move from the peripheral nervous system, so from the PNS to the central nervous system. The next classification is a motor neuron. It follows an efferent pathway. It is the efferent neuron, meaning it leaves. So it's going to go in the opposite direction from the central nervous system to the peripheral nervous system. And then the next are go-betweens, are interneurons. They are the go-betweens, the interpreters, the one that's interpreting information from the sensory neuron and then converting it into some type of response for the motor neuron. So again, they are the go-between. They are the one that's taking that information, that stimuli, that sensory information coming in and saying, hey, what is this? How do I respond? And then once it decides what the response is, it sends it via a motor neuron through that motor pathway. So let's put it all together into a picture to explain how it happens. So we start here. 1. Sensory receptors detecting a stimulus, some type of, let's say, crude touch or constant pressure. And then it propagates a signal, a nerve impulse down. Look, this is a unipolar neuron, right? Okay, so propagates a signal down the unipolar neuron from the peripheral nervous system into the central nervous system, for which we have a multipolar interneuron, our go-between. Right? And again, look, interneurons are typically multipolar. So this is translating that sensory signal into a motor response. Now this talker here, the synaptic M bulbs, remember we refer to them as talkers, whereas the dendrites are listeners. And this is a good example to say, well, this interneuron, the listeners are listening for that sensory stimuli, that sensory information, and translating it into, so talkers are translating it into a motor response. And then that multipolar motor neuron will process a nerve impulse, an action potential, to an effector muscle or gland that will elicit the response. Our next subset of nervous tissue are neuroglia. Now, what do we know about neuroglia? They are different from neurons because they are not electrically excitable. They make up a little more than half of the volume of the nervous system. They have the unique ability to multiply and divide. And we find 6 distinct types of neuroglia, 4 in the central nervous system and 2 in the peripheral nervous system. So before we get into the specifics of the neuroglia, let's look at a picture just to see some few different types. We have ependymal cells. We have oligodendrocytes, protoplasmic, don't worry about the protoplasmic, but astrocytes. We have microglia. We have some more microglial cells here, oligodendrocytes, another astrocyte, protoplasmic versus a fibrous astrocyte. We're not going to get into the differences between that, but notice that they are around, supporting, attached to, insulating the actual neurons that we just got finished talking about. That's a key component. So let's outline and discuss them. The 4 types of neuroglia found in the central nervous system. The first are ependymal cells. These cells line the hollow regions of the central nervous system, specifically the ventricles of the brain, and the central canal of the spinal cord. Their job is going to be to help produce and circulate cerebrospinal fluid. The second are astrocytes. Now, their name tells us they're star-shaped. They are large cell bodies with many processes. These are the most numerous of our neuroglia. They can help guide axon regeneration in our peripheral nervous system when they get damaged, cut, etc. But they're also really important in making what is known as our blood-brain barrier. They are protection for the cells in the central nervous system. The third type is an oligodendrocyte. Oligodendrocytes form the myelinated sheaths for the axons of the central nervous system. So if you go back a couple slides to that previous picture, and we saw that oligodendrocyte having this extension, this process, and wrapping, forming a grip, a fat coating, a myelin, this myelinated sheath around the axons of the central nervous system. Again, now we're in the central nervous system, not the peripheral, so that's a key difference. In the peripheral nervous system, they're called something a little different. The next are microglia. These are phagocytic cells. They're small and the least numerous, and they function as our little Pac-Men, our macrophages. Now we can switch gears and look at the two distinct neuroglia found in the peripheral nervous system. The first are called Schwann cells. Their job is to myelinate the axons of the peripheral nervous system. They also help clear cellular debris when we require regrowth. So if you were to get a cut and it cut through the axon of a sensory neuron or motor neuron, the Schwann cells helps to clear the debris of that broken myelin for regrowth. Now remember that myelin is really just fat. This is a great insulator, so we're going to use it as an insulator of the axon. And we'll talk later on when we look at action potentials why these insulators are super important. The next are satellite cells. They surround neurons. They're similar to the astrocytes in the central nervous system, and functionally they contribute to chronic pain. So if you've ever had a burn in your skin and you have that throbbing chronic pain, typically the satellite cells are contributing to that. So our flow chart here talks about all those different neuroglia that we just outlined, and specifically where they're found and how they operate. So for example, we can start with satellite cell. We see that in the peripheral nervous system, they surround neurons. In the ganglia, they regulate oxygen and carbon dioxide, nutrient and neurotransmitter levels around neurons in the ganglia. The next is the Schwann cell. So there's our second neuroglia found in the PNS. So it surrounds all axons in the peripheral nervous system, and that's key, right? So Schwann cells are only found in the peripheral nervous system. In the central nervous system, they're different. It is the oligodendrocytes that contribute to that myelin. So their job is to participate in repair of processes after injuries and insulating the axon of a multipolar neuron. In the central nervous system, we had that four. Remember, our ependymal cells line the spaces, the ventricles of the brain. This is deep within the cerebrum, the central canal of the spinal cord, and produces cerebrospinal fluid. Our microglia, these were our phagocytes, so remove cell debris, wastes, and pathogens by phagocytosis. Our star-shaped ones, which were our astrocytes, setting up the blood-brain barrier, providing structural support, regulating ions, nutrients, and dissolved gas concentrations. They also absorb and recycle neurotransmitters, and when damaged, they form scar tissue after injury. The fourth and final are our oligodendrocytes. These were the myelinated their job is to myelinate the axons of the central nervous system, and they provide a structural framework. Let's examine the myelination of the neurons. The myelin sheath is produced by Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system. It surrounds the axons of most neurons. So at any one of these pictures, you can see the development of the myelin sheath. Again, this fat that is surrounding. So if you look down here, look at all this fat, the myelin that is surrounding the axon. So from development, we get the nucleus and the Schwann cell that kind of engulfs and wraps around and starts just rolling and rolling and rolling and rolling around, forming this. You can see here, if you were to follow that arrow, we're winding our way around here, forming this myelin sheath that we see here. Again, we look at unmyelinated axons in our picture over here on to the right. So we'll label this 1 and over here 2. And you can see that we have Schwann cells, the cytoplasm of Schwann cells, surrounding all these unmyelinated axons. And again, this is a transverse section. We're cutting through it, not looking the length of it, of myelinated and unmyelinated axons. So the formation is pretty cool. It's pretty important. It's similar to what we saw in bones, right? When we talked about growing bones widthwise, and we talked about like a cinnamon roll type of orientation. So here it is, that cinnamon roll again, just rolling around, layering that fat on top of that axon. Now we can take those myelinated and unmyelinated neurons, bundle them together, and talk about how they look in the central nervous system. So we call this gray matter and white matter. Now, the gray matter is called gray matter because they are those unmyelinated axons. The white matter is white because what you're seeing is all that fat, all that myelin surrounding the axon. So myelinated white, unmyelinated gray. Now, interesting note, look at the spinal cord. The gray is on the inside and the white is on the outside. Now, when we get to the brain, it reverses, right? So the gray is on the outside and the white is on the inside. So that's an interesting note to pay attention to as we talk about and identify the anatomy and physiology of these structures. Now that we have talked about the anatomy of neurons and neuroglia, our next step in the nervous system is to discuss electrical signals in neurons. How do they propagate? How do they develop? And what do they do? Electrical signals in neurons. Excitable cells communicate with each other via action potentials or graded potentials. So in our next few slides, we're going to discuss and differentiate the difference between these two. So action potentials allow communications over short and long distances, whereas graded potentials allow communications over short distances only. Production of an action potential or graded potential depends upon the existence of a resting membrane potential and the existence of certain ion channels, all of which we have outlined in our previous lectures. So let's take a look at how a complete graded potential gives rise to action potentials. Now remember, these are summative, so they have to summate enough to fire that action potential. So assuming that the stimuli at the graded potential, the sensory receptor, are strong enough, it will fire and trigger an action potential. So we start here with number 1. Our sensory receptor is detecting the pencil or pen in our hand and sending a action potential. So this is a graded potentials down through the hand, through the arm, into the spinal cord, into the dorsal root ganglia. So this is a dorsal root ganglia, DRG. You notice it's a unipolar sensory neuron. And into the brain, where we have our go-between, our interneuron. Now that interneuron is going to send the information to the cerebral cortex, which is going to fire more action potentials down the motor pathway, which is an efferent pathway out the ventral root. And that's going to go to the muscles, the skeletal muscles in the hand. That's going to fire an action potential to get those muscles to contract. Again, go back to our lecture on the neuromuscular junction to understand how those the action potential of the NMJ gives rise to the sliding filament theory, the contraction of those skeletal muscles. Now we have to outline those transmembrane potentials. We have 5 main membrane processes, the first being resting membrane potential. We're going to assign this a number of about minus 70 millivolt. So this is the potential, the charge on a cell when it's at rest. So if you were to put a voltometer into the cell membrane, you would get inside that cell a resting charge of about minus 70 millivolt. Now, does this mean that there's a lot of negatively charged stuff in there? No, it's the distribution of positively charged ions that weighs it higher or lower as compared to the outside of the cell. Next is a graded potential. These are temporary localized changes in resting potential. It's caused by a stimulus. Now, an important note is right here. A single graded potential will not initiate an action potential. Remember, we said that we have to summate these in order, so we're going to add all these graded potentials that will give us a point of threshold, so a specific number, a charge that's going to allow that action potential to fire. Next are our actual action potentials. This is an electrical impulse. It's produced by depolarized graded potentials that are summed. So again, keywords. We're depolarizing parts of a cell, which are our graded potentials, that are going to summate or add them together to bring us to a specific threshold number that's going to fire this action potential. Now, another key point about action potentials is that they occur in an all or none fashion. So they either happen or they do not. Again, it depends on if the summation of these graded potentials can reach threshold, which will allow, so we'll write threshold here, to reach threshold. We're going to assign numbers to these. Reach threshold to allow for that all or none principle to occur. Next, the action potential will propagate along the surface of axons to the synapses. Number 4 is our synaptic activity. So again, go back to that neuromuscular junction. Remember those synaptic end bulbs. What's happening there? We're releasing neurotransmitters at a presynaptic membrane into that cleft, right? That synaptic cleft. It's producing graded potentials in a postsynaptic membrane. So again, here we're talking from neuron to neuron. We could also put this to neuron to muscle, like the neuromuscular junction, but we still have a presynaptic membrane and a postsynaptic membrane. Next and last for our 5 main membrane processes, number 5 is information processing. So what happens? Response of that postsynaptic cell. So how does that postsynaptic cell respond? And if the intensity of the depolarization spreads to the hillock, then threshold is reached and an action potential will occur. Now, if the intensity of the depolarization doesn't spread to the hillock, the threshold is not reached and an action potential will not propagate. So again, this is that, we're going to put this all or none idea, right? That all or none principle. The next step in our process is to discuss ion channels in neurons. So how do we allow for a change in conductivity of the cell? How do we allow for the cell to become depolarized, repolarized, hyperpolarized, and then again maintain a resting membrane potential, right? That minus 70. So remember before we said all we're doing is simply distributing charges differently from the outside and the inside. So let's take a look at these 4 channels. So the first of our 4 channels, we'll number this number 1, are leak channels, and their name tells us exactly what they are. They're ATPase-gated channels. They leak sodium and potassium in and out of the cell. Now, potassium channels are more numerous than the sodium channels. So we'll see that when our potassium leak channels are closed, potassium is maintained within and outside the cell. Now, it will physically bebop, open and close to allow for potassium to leak out. Again, what these do is it maintains that minus 70 resting membrane potential. That's what leak channels do. They allow for any types of charge disruption, so if the cell's moving something in or out, these leak channels will open and close, allowing sodium and potassium to move freely, again, maintaining that resting membrane potential. That's their job. The second are, we'll write number 2, are our ligand-gated channels. So these are responsible for binding a ligand to the receptor, which in our case, we're going to talk about acetylcholine, remember the most predominant neurotransmitter. So when acetylcholine is present, it binds to this channel. The channel will open and allow for stuff, calcium, sodium, chloride, to move in or out. So again, let's look what's going on here. Here comes our acetylcholine. It binds to it. The channel physically opens, and we have potassium, sodium, and calcium moving in or out to the outside extracellular fluid or inside into the cytosol. Now the next question is, where do we typically find these channels? So we're going to find these along the dendrites, the cell body, the soma of the neuron, right? And this is what's going to give us, so ligand-gated channels are responsible for those graded potentials. The third type of channel is mechanically gated channels, which respond to mechanical vibrations or pressure stimuli. Typically, we find these in our skin. Okay, so with stretching of our skin, pressure to our skin, The stretch of the cell membrane will open the channel, allowing for calcium and sodium to move into the cell, again allowing for an increase in conductivity. So if you think about resting membrane potential as being minus 70 millivolts, you can imagine as you start flooding in positive ions, this charge will change and will increase and go to minus 60, minus 50, all the way up that scale. The fourth are voltage-gated channels. Now we have two types of voltage-gated channels. We have sodium and potassium. And we're going to talk about these and outline these in a little bit more. Now if we look at sodium channels, we'll give some dynamics of it. They are fast to open and fast to close. I'm introducing this now because I want you to keep this in the back of your mind as we're talking about action potentials. Now, for potassium channels, they're the opposite. They are slow to open and slow to close. Again, keep this in the back of your mind because this is going to be super important as we talk about the dynamics of these channels, when they open, when they close, when they inactivate, and what that means for the curve of and the states of these cell membranes, depolarized to repolarized to hyperpolarized. So voltage-gated channels respond to direct changes in membrane potential. So that means they will open and close on a specific charge. For example, for our scenario, and they're all different throughout the Bible, but we're going to kind of standardize it and say for our sodium channels, these guys are going to fly open at about minus 55 millivolts. That's going to be our threshold. Our potassium-gated channels are going to open at plus 30 millivolts. So again, this is just opening. We're not really covering anything else yet, when they close, when they inactivate, etc. So table 12.1 goes through the different types of channels, the four types of channels: leak channels, ligand-gated channels, mechanically gated channels, and voltage-gated channels. Where you typically find them, so locations. Remember, our leak channels are found nearly in all cells, including dendrite cell bodies and axons. Again, they're maintaining resting membrane potentials. Our ligand-gated channels, remember we said they were by the dendrites and the soma of the cell body of these neurons, and pain receptors and dendrites and cell bodies of interneurons and motor neurons. Again, these are contributing to those graded potentials that will give rise to a action potential. Mechanically gated channels, again, the description we talked about: location, dendrites of sensory neurons such as touch, we said touch and pressure, and some pain receptors. And then look at our voltage gated channels, localized, so to the axons of all types of neurons. So again, this is our action potential one. So if we separate and kind of put these together, and if we were to draw, I'm going to draw a quick neuron for you here, and we have all our dendrites, and we'll get this drawing a little bit better. We talk about this region, the cell body, as being graded potentials with those ligand-gated channels, so ligand-gated channels, and then the axon is going to be our action potential with our voltage-gated channel. So that kind of puts it together. Again, we're going to look at this again in our next slide, so it'll make a little more sense. So now we're going to spend some time elaborating on each of our potentials: resting membrane potential, action potential, graded potentials. So let's start with the resting membrane potential. So the membrane of a non-conducting neuron is positive outside and negative inside. Remember, we said that resting membrane potential, we gave it a number of minus 70 millivolts. And again, RMP of cells vary throughout the body. We're going to use a common number just to align everything and make it a little more easier. Now, how do we determine this minus 70 millivolt resting membrane potential? So there's an unequal distribution of ions across the plasma membrane and the selective permeability of the neuron's membrane to potassium and sodium. Now, most anions cannot leave the cell, which is why we need those leak channels, those ATPase-gated sodium-potassium pumps, which is going to help to maintain that minus 70 millivolts. Now, there are three requirements for resting membrane potential. Number one, the concentration gradient of ions, so sodium and potassium. Number two, it has to be selectively permeable through those leak channels that we just talked about. And number three, we're going to maintain charge difference across the membrane. We're going to maintain that resting potential at minus 70 millivolts. Now let's look at the transmembrane potential with regards to resting membrane potential. So these are passive forces acting across the membrane. So remember our first week of class we talked about electrochemical gradients? Well, here we're going to elaborate on it. So we deal with chemical gradients, so the concentration gradients of ions, specifically sodium and potassium. Now the electrical gradients are separate charges of positive and negative ions which could result in potential difference. So we can put it to drawing. So if we look at the first one here, we look at a distribution of charge that produces the resting membrane potential. In the extracellular fluid, right here, we have an equal number of positive and negative charges, which is typical in most ECFs. Now for resting membrane potential and electrical potential differences across the plasma membrane. So again, we're more negative inside than we are positive. Now for the cytosol, here, we're dealing again with an equal number of positive and negative charges, which is again typical in most cytosols. Now let's put a voltmeter in and measure these differences. So number 2 is going to be the measurement of the RMP, resting membrane potential of a neuron. So we stick that electrode into the cell, and what do we get? More negative. So see all these negatively charged. So these aren't negatively charged ions. This is the charge difference. So again, more negative inside where the outside is more positive. Okay, so the difference, again, look at our charge. We're at minus 70 millivolts. So that's a good resting membrane potential for the cell. Now we can look at another picture to help us with the dynamics of resting membrane potential. So again, we see that there are some negatively charged proteins that are going to help contribute to that resting membrane potential, but more often than not, it is the distribution or redistribution of these positive and the few negatively charged ions. Again, we're specifically talking about sodium and potassium, so positively charged ions moving in and out of the cell. So what do we see here? We see the sodium potassium ATPase gated pumps. So here they are. Notice we need ATP. They're going to power this pump to cycle in potassium and sodium. Now look at the distribution. They're going to be about a 3:2 ratio. So for every 2 potassiums leaving, 3 sodiums will be coming in. So again, we see 2 potassiums going out. We have, with our ATPase pump channels, our leak channels, 2 potassiums coming in. We have 3 sodiums moving out. So factors contributing to the RMP, or resting membrane potential. So we see a lot of positive and negative stuff along here. So what do we see in the extracellular fluid? We see sodium ions, positive, potassium ions, positive. Look at the distribution, more sodium than potassium in the ECF. But we do get a bunch of these chloride ions also distributed in the extracellular fluid. Now these will contribute to the hyperpolarization or an inhibitory type process that we'll just talk about a little later. Now if you look in the cytosol, we see a lot of potassiums, a lot of positives, some sodiums. Again, this is a typical distribution. We have our negatively charged proteins, and then we have a few negatively charged phosphate ions. So again, this is going to help set up our resting membrane potential. Now we also see a couple leak channels, potassium leak channels, sodium leak channels, and some ATPase gated sodium potassium channels. Again, all contributing to resting membrane potential. The last thing we're going to look at regarding resting membrane potential are the active forces across the membrane. Now, we alluded to, now we're going to outline those sodium potassium ATPase gated channels, these pumps. They have 4 main characteristics. It is powered by ATP, so we need energy here. So we need energy in order to power these pumps. It carries in a 3:2 ratio, 3 sodiums out, 2 potassiums in. Its goal is to balance the passive forces of diffusion. Its ultimate goal is maintaining resting membrane potential of 70 millivolts. So we're going to maintain resting membrane potential. The next grouping we're going to look at are graded potentials. So, what are these? Remember we said they were localized to this region, the cell body of the neuron. Now, what are they? They're small deviations in resting membrane potential. And again, if we summate them, and if they're large enough, they can give rise to action potentials. Now, let's define our graded potentials. They are also called local potentials. They produce changes in transmembrane potentials. They may be depolarization where inside of the cell gets more positive. So again, we're talking about depolarizing the cell membrane over short distances. So this is key that we're talking over short distances, only the cell body. Right? They may be hyperpolarizations where inside of the cell gets more negative. So now we're getting real specific. And if it's depolarizations, we're going to call them EPSPs for excitatory postsynaptic potentials. Now, if it's hyperpolarizations, we're going to call them IPSPs for inhibitory postsynaptic potentials. And if you're alluding, if it's excitatory, this may give rise to an action potential, whereas inhibitory postsynaptic potentials will not give rise because it is preventing, it's inhibiting the depolarization of the cell by hyperpolarizing it. Now, lastly, for graded potentials, a single graded potential doesn't spread very far into the cell body, which is why we have to summate them. So this is why we have to summate them up. Summate them. So now a graded potential occurs in response to the opening of mechanically gated or ligand gated channels. Now, we're not going to focus on the mechanical, we're going to work on ligands. So we're talking about these guys right here. Now, what happens? Here comes our ligand. Remember we said our ligand is acetylcholine, the most predominant neurotransmitter in the body. Now, this is an excitatory neurotransmitter. So what does that mean? Excitatory. That means that this is going to excite this cell, allowing for depolarization to occur. So this is typically what we want for action potentials. Now the second one on the bottom here, if you notice, is allowing for a ligand called glycine to attach to this channel, which will permit chloride ion. Now chloride's negative, so that means this is an inhibitory neurotransmitter. Next, we're going to look at stimulus strength and how it contributes to these graded potentials. So let's look at the amplitude of a graded potential and how it's going to depend on the stimulus strength. So our first stimulus gives us this little amplitude, and remember that our goal is to get to minus 55 millivolt, which will fire that action potential. Now the first stimulus only went to minus 65, so that didn't give us didn't allow us to reach threshold, which didn't allow the action potential. Now stimulus 2 has a little larger of an amplitude, and it went to about minus 62 millivolts, still not enough to bring us to threshold. Now our third stimulus, albeit still even greater in amplitude, brought us to about minus 60. So again, still not enough to bring us to threshold, but again, we're getting there. Now the next key will be to summate the stimuli to allow for a larger graded potential to bring us to that threshold mark. Let's summate these graded potentials. So we're going to call it summation. Now those graded potentials can be added together to become larger in amplitude. So let's see where we fire our stimuli. Stimuli 1 brings forth a depolarization event, and then before it's able to come back to resting membrane potential, here comes stimulus 2, firing another depolarization event. And as you can see, we're slowly getting up to that minus 55 millivolt. Now if we bring another stimuli here, and let's elaborate on this table, say stimulus 3, that would give us another incline closer to that minus 55, and then stimulus 4 would take us up and over. So again, summation is key with graded potentials to get us where we need to be for those action potentials.